On we go with the rest of the chapter. I just poked myself in the eye with my glasses there. Uh, okay, so we left off talking about the epigenetic business here. And now we want to get into the development of the structures of the nervous system. So, as I mentioned before, we have these stages with neurogenesis, followed by neuromigration, and then synaptogenesis. We'll get into that a little bit here, but what we're also going to need to talk about here is the formation of the larger structures. You already know the basics, right? From one of the earlier chapters in the book, you know the organization of the central and the peripheral nervous system, and the division of the central nervous system into the spine, plus the brain stem that includes the hindbrain and midbrain, as well as the forebrain and all of the forebrain structures. We see an early version of this here. So this is part of the embryonic stage of development. So this starts around 3 weeks after conception, right? So conception is when we're a single cell fertilized egg. 3 weeks in, as I had mentioned in the previous video, we're going to see the formation of the three layers: the endoderm, mesoderm, and ectoderm. And the ectoderm is going to start forming into the skin and the rest of the sensory organs, as well as the nervous system. And so we're going to see as that starts to fully develop, the nervous system forms. And so we see labeled here as the in red, the neural tube. And the neural tube notes here that's going to form into the spinal cord, but ultimately the neural tube is where a lot of the neurogenesis happens. At the tail end, you can see the tail there forming. Obviously, we don't have tails, so eventually that's going to be kind of enveloped by the developing lower limbs of the body. You can start to see here some limb buds that are forming, right? Just a little small. I think you might be able to see my mouse on the video here. Limb buds are basically the very early arms that I'm pointing to here and legs down here. Eventually, as those develop and get bigger, the sacral end of the spine, which is where the tail end will sort of become kind of like I said, enveloped with the development of the lower limbs. Um, we can see the endoderm is not really highlighted here, but the organs are forming where I'm pointing here with the mouse. So we see the hindbrain structure in purple, the midbrain in green, and the forebrain in yellow. So if we think of the brain in totality here as all three of those parts, that represents a pretty large part of the nervous system at this phase of development. Like I said, this begins at 3 weeks in, but this picture is probably capturing something that's a little bit further along, maybe closer to 4 weeks or so. Um, but this is illustrating an important point. The idea is that all three of these structures that represent the brain itself are approximately, about a third of the total length of the brain at this point, right? So in this situation, we're seeing that the brain is developing before a lot of the rest of the body is, right? We're putting a lot of energy into growing brains before we grow any other part of the body. And this is an important developmental principle called the cephalocaudal principle. Cephalo means head, caudal means tail. And so the idea is that development goes in this head to tail direction. And that's what we're seeing here is the development of the brain, and the head is going to take precedence over everything else. A lot of that is important for our class because that's going to be relevant to understanding why we have the brain sort of ready to go at the moment we're born. Because then when we look at how the brain develops before we're born versus after we're born, we're going to need to focus on the importance of development or of experience, I mean to say, right? So experiential factors in early childhood are going to have a huge influence on the organization and function of the nervous system. But that nervous system needs to be ready to go at the point where we're born. So it's ready to start processing those experiences and responding to those experiences. So we have to put a lot of energy into growing these brain structures before we get there, right? So we see that. Now, notice, of course, that at this point, the forebrain, which will develop into the cerebral cortices, is not yet quite so big. That's going to take a lot of time before it really starts to look like a fully formed brain. Um, So let's kind of break it down here. So as noted, we're going to say that the period of the zygote is the first few weeks here from the fertilized egg all the way to the end of the second week. And once we get into the start of the third week, we now move into the phase known as the embryonic stage of development. This is when we see the cell specialization happening, right? So in the period of the zygote, we're looking at mostly these undifferentiated stem cells, but when we go through this process, from the totipotent to pluripotent to multipotent, right? That's when we see that now these embryonic layers. And so between the third week and the eighth week after conception, that is this phase called the embryonic stage when all of these organ systems are going to start forming for the very first time. And that is really the definition of the embryonic stage is the appearance and emergence of all of the different organ systems. From 9 weeks on to full term is the fetal stage. And here, everything is really just about the development and maturation of all of those different organ systems. We're going to focus here again on the nervous system. So we're going to start with what's called the neural plate. And then we're going to need to turn the neural plate into the neural tube. And maybe it's better for me to go to the pictures here. This is going to be a theme in a lot of my different slideshows is that I've got text on one screen and pictures on another screen that kind of go with each other, right? Top left here, 18 days in, right? So we are right at the end of that zygote stage and into the very early part of the embryonic stage. And so we see the neural plate forming, right? And so then it's called the neural plate because it's just generally a flat layer of tissue. Highlighted in red here is this deep layer called the mantle layer. And the mantle layer is exactly where neurogenesis is happening, right? These stem cells are rapidly dividing and becoming multipotent neural stem cells at this point, right? Move to the right and top right. And so we start to notice a couple of things is that the neural plate starts to sink a little bit and become a neural groove. And one of the key themes of why we want to do this is this increases the surface layer of the entire neural plate area so that we have more cells and we have room for more cells and room for new things to more of these stem cells to be dividing. But one of the things you also might notice here is that right at the middle of the neural plate or neural groove, we're starting to see it close, right? And if we go down to 22 days, sorry, in the middle left, we're starting to see it actually fully close up in the middle. And then that closure is going to start moving towards both the head end and the tail end here, right? So now instead of a neural groove, we're going to call it a neural tube. And the neural tube is really the spinal cord, right? And so it has a hollow fluid-filled cavity in the middle full of cerebrospinal fluid. But that closure begins happening there in the middle of the spine. So we're going to zip it up essentially, right? It's just like imagining a zipper working in both directions. And so it needs to zip down towards the tail end of the spine. That should happen normally. If it fails to close at the tail end, that results in a birth defect called spina bifida. Can be fairly serious in some cases, can be pretty mild in others. Um, but on the head end, it's a bit more catastrophic if it fails to close on the head end. So we're going to zip everything up head to tail. We're going to close up the neural tube entirely at this point. It takes a few days. As you can see, by 24 days, we have a fully enclosed nervous system here. But again, deep into the mantle layer of the neural tube, we're going to have extremely rapid neurogenesis, followed by neuromigration as these brand new neurons migrate to start developing the brain bit by bit. I have a little more detail on what's going on with these cells here. So we start with the neural stem cells, as I said. These are the multipotent stem cells, right? So they are multipotent because they've already become slightly more specialized compared to the original totipotent or pluripotent stem cells, but they've been restricted a little bit. So here the restrictions mean that they can become neurons, and there are different kinds of neurons, right? And the neurons that make up, say, sensory neurons or motor neurons or interneurons in the brain or spine, those have a different structure. It's also the case that neurons in the, say, the cerebellum have a particular structure that makes them very different than the neurons in the cerebrum. And, it's worth pointing out too that neurons in, say, the hypothalamus versus the amygdala versus the visual cortex, they have different kinds of receptors and different kinds of neurotransmitters involved in their activation. So there is a further need for much more fine detailed specialization that's going to happen here. These multipotent cells can also become glial cells, as I had noted before, right? And there's different kinds of glial cells too. So all of those different specializations have not yet happened, right? They are still multipotent cells. So that means that they do have some future possible potential developmental changes that can happen to them. One example of this here is a progenitor cell, right? So the progenitor cell derives from the stem cell, the multipotent stem cell. That's going to migrate, right? So when the cells are migrating, they are still progenitor cells at this point. Very fairly simplified cells, just a cell body with no specialized structure. Then once they get into place, that's when we're going to activate the particular gene sequences that will start generating the appropriate proteins that turn it into the appropriate kind of cell that it's supposed to be. Right? It's going to start doing the appropriate metabolic functions associated with a neuron or glial cell or whatever it's supposed to be at this point. The immature neurons are actually called neuroblasts. So once the progenitor cell specializes to become either a neuron or glial cell, it's going to become a neuroblast if it's going to become a neuron. And the neuroblast can then further specialize to be the appropriate kind of neuron if it's going to be a neuron of the, as I said, the cerebellum or cerebrum or hypothalamus or whatever it might be. And then likewise, if that progenitor cell is going to develop into a glial cell, let's say like an astrocyte, because astrocytes an important part of the brain that form the blood-brain barrier and help extract nutrients from the blood vessels and regulate the metabolic function of the neurons. Then that's happening there with those progenitor cells turning into glioblasts, and then they can further become different kinds of glial cells. Other kinds of glial cells would be things maybe like a Schwann cell or an oligodendrocyte that help myelinate neurons, right? They help form the myelin sheath around the axons. So some of these processes we've been talking about already, right? So we're going to have cell births, right? Cell creation. We've got neurogenesis, we've got gliogenesis now all happening. As I've said, we're going to do the majority of this prenatally so that we're born with some large percentage of all of the neurons, these 85 billion plus neurons that we have. Even, in fact, a lot of it happens even by 5 months gestational age, right? So typically, of course, we're looking at 9 months of gestation, but a whole big bunch of that neurogenesis happens in those first 5 months. It slows down. It doesn't stop, but it does slow down from 5 months to 9 months there. There are, of course, though, places in the brain where new neurons are made for the rest of our lives. And an important area of this is the hippocampus, right? The hippocampus, again, much later in the semester, we'll talk about how learning and memory work. But the hippocampus is an important part for embedding new long-term memories. So acquiring new memories requires new connections in the brain and even new neurons. And so we're going to need to have some of that happening in the hippocampus and some of the surrounding regions like the entorhinal cortex and parahippocampal cortex are going to benefit from having some new neurons across the lifespan. We've talked about cell migration already. So I said that glial cells are going to provide a pathway here. This is a specific kind of glial cell called radial glia. And they're essentially building these pathways that I've compared to ladders or a lattice work. And what they do is they emit different kinds of chemicals. And the progenitor cells that are doing the migration, they're going to sniff out different chemical trails because there are going to be a variety of different chemical trails that help these neurons know which path they need to follow, whether they're going to become part of the brainstem or part of the cerebellum or part of the hypothalamus or what have you, right? So they're going to follow different pathways to get to those appropriate parts of the brain so that once they get into their so-called final destination, then they can start to differentiate as we have here, right? And this is where we're going to see this phenotypic expression, right? Phenotypic expression refers to the idea that we're going to see this epigenetic expression where we have to start manufacturing and synthesizing certain neurotransmitters Well, which ones? Is it going to be dopamine, serotonin, GABA, glutamate, right? Different neurotransmitters have different functions in different parts of the brain. So the neurotransmitter has to express the appropriate phenotype, that is, to start making appropriate neurotransmitters. And it's also going to have receptors on its dendrites that will respond to certain kinds of neurotransmitters. So that again means unique receptor types on its dendrites. After the differentiation, now we see a form of maturation, sorry, called arborization. So you might notice that the word arbor sounds like trees, right? Arbor Day. Well, that's exactly what the researchers are using as an analogy here, right? We're seeing this branching occur. In fact, the word dendrite itself, I think, is derives from Greek. Don't quote me on that. I always forget, but I think it's Greek for tree branch or something like that. So the fact that you see this increasing branching between the neurons, right? So they get into place and they just have a simple structure, but they start growing more dendrites and longer axons, and then they start interconnecting with each other. And this happens in, a little bit prenatally, but we start on the left with a brand new, baby, newborn. And by 1 month and 3 months, we see a little bit of arborization, but we continue to see that going all the way out here to 24 months, 2 years, a lot of extra new branches. So the brain continues to grow, right? The brain's going to get bigger here and extra mass, but that mass is not necessarily a lot of new neurons as much as it is just the neurons themselves are getting bigger because they're growing longer axons and more dendritic spines. Here we have a video that can capture a little bit of this. Let's see if we can get this to work here, video and video. Um, so you have here just some very immature neurons that are starting to branch out, right? They're starting to grow these little axons, and the axons, keep in mind, axons have small little branches at the end of them too, right? The axon terminals. And you see them actually moving and finding others. And once they find another neuron, right, then they start forming connections here. So this is called synaptogenesis. So as I've mentioned already, we have somewhere in the ballpark of 85 billion neurons. How many synapses do we have in the brain? Well, it's a big number, right? 10 to the power of 14 is a extremely large number. Keep in mind, no one has ever sat down and counted 85 billion neurons or 10 to the 14th synapses. These are estimates. Um, but, we reasonably think those are accurate estimates here. So then what happens? We're going to get rid of neurons and synapses. That sounds somewhat ironic, right? Why would we want to do this? Well, part of the issue is, so I've got more pictures down here of this arborization process again, right? So this time it shows some arborization happening even prenatally, right? 36 weeks gestational age, which is a couple weeks short of full term. Then we have a newborn, 3 months, 6 months, 2 years, right? So we see the increasing arborization, but notice at 4 years and 6 years, things look a little sparser, right? Why do we do that? Why would we start to eliminate some of the synapses that we have been building over those first 2 years of life? So, what is happening here is that a lot of this synaptic formation is happening in a way that we might say is blind, if that's maybe an appropriate way to think about it. So, we need synapses for a reason, right? What do we need synapses for? Well, we need synapses for everything. Let's use an example. A good example is vision. I need synapses that allow me to see. I'm just going to grab a pen here. In order for me to recognize this, to see it, to be able to process its size and shape and color, I need synapses in the occipital lobe of my brain that allow me to process the visual information for that. And if I don't form those synapses correctly, then I'm not going to have appropriate visual function, right? I need synapses for language as well. And so the idea here is that how is it that I develop the ability to have the right synapses that are needed to see the colors and the size and shape of this pen? Does my brain possibly know in advance, maybe encoded into the DNA, which synapses are needed for that visual ability? And the answer is no. As you saw in that video, it doesn't look like that synaptogenesis was driven by some sort of predetermined mechanism. It was just as if the neurons were just kind of exploring and branching out and hoping to hook up with some other neuron and then find that connection and then start forming these connections, right? So the brain is not going to be able to quote unquote know in advance which of those synapses are useful for seeing the color blue versus the color red or any of that kind of stuff. It's just going to require uh, essentially, I don't want to say luck, but rather it's going to require the over proliferation of synapses, right? We're going to form way too many synapses. We're going to form more than we really need. And then what happens is that experience occurs, right? And so I'm going to look at some things as a baby and I'm going to see colors. And the colors that I'm exposed to will use some of those synapses that have been forming. And therefore, I'm going to strengthen those synapses by using them, right? This is a use it or lose it kind of phenomenon. And this is what I mentioned in the previous video is famously called Hebb's Law. What happens is that by using those synapses that are important for seeing the color red or seeing the color blue, I'm going to strengthen them. Those synapses will stay alive, essentially. But there will be lots of other synapses that have formed in my brain that don't get used for various reasons. They just turn out to not be functionally relevant for a lot of typical experiences, like looking at the kinds of objects that a baby might see every day. Language is a similar kind of an issue here because when we think about human language, we think about the fact that there are lots of different languages and they're different from each other. There are lots of regularities and universals when it comes to human language. Maybe the grammatical structure of language has some universals, but obviously the vocabulary of the language differs and there's a few syntactic differences between language. There's also a lot of sonic differences between languages, right? There are phonemes, sounds used in some languages that are not used in other languages. There's no way for your brain to genetically know whether you're going to be born into an English speaking household or a Spanish speaking household or a Mandarin speaking household or any of that sort of stuff, right? Your brain's not going to know what language you're going to be hearing when you are a baby. So you don't have this ability to form synapses that are relevant for processing the sounds of English versus processing the sounds of French, right? Um, but rather, what happens is that you over proliferate so that your brain is ready to process any possible sound, and especially the sounds of language. And so, what we see is that newborns are especially sensitive to any possible human speech sound, and they can easily hear and distinguish between a lot of different kinds of phonemes. But then what happens is that over the course of their first year of life, they're going to typically be exposed to just one language. Maybe in a bilingual household they'll be exposed to two, but that's typically the max there. So one, maybe two languages. And so again, there's a subset of phonemes that are going to get activated, not phonemes, sorry, synapses from hearing those phonemes that will be activated. And that's going to strengthen those particular synapses. But then there's going to be a whole bunch of other synapses that might be useful for other languages, but they will never get activated because the child never gets exposed to those sounds. And so then we're going to start pruning them away, right? It turns out that they're going to be unnecessary given this child's experience over the first year or two of their life. So it's time to get rid of the unnecessary synapses, right? So this is the idea. We over proliferate, right? This is what I mean at the top here of overabundance. We have more neurons, we have more synapses than we really need because we don't know which ones are exactly going to be the ones we need. So we generate a bunch and then we start having experiences. And this is what I said. This is why we want to build all this stuff before we're born so that way we're ready to start processing these experiences. And then experience determines which connections are useful and which ones are not useful. And this is going to take a few years, right? It's going to take 2, 3, 4 years. But then the idea is that once we've determined this, we can now afford to start pruning away the excess, right? The ones that are not needed. And this is called synaptic pruning, right? So we're just going to say that, okay, this particular dendrite that's connecting to this axon over here, we don't really need that anymore. So let's just get rid of that dendrite, right? Well, let's just let it atrophy and get rid of it, or disconnect it from that axon and connect it to a different axon. Likewise, some neurons in the brain never actually receive any synaptic input from other neurons, right? They end up just basically not being used. And when a neuron is not stimulated by any synapse, what happens to it? It it kills itself, right? This is called apoptosis, and it's a genetically programmed process where a neuron essentially just dies from lack of stimulation. So that's what's going to happen here is we're just going to get rid of unnecessary neurons. We're going to prune away a bunch of unnecessary synapses, and we're essentially fine tuning the brain at this point. We're not going to waste any metabolic resources keeping those connections alive if they're not needed. So we're going to sharpen everything up, right? Now, I've compared this here to an evolutionary process, right? The concept is called neural Darwinism, with the idea that these neurons are competing for metabolic resources, and so we only need to keep the ones around that have value, right? That have survival value, right? So we prune away the unnecessary ones, and we keep the ones that have the appropriate um, characteristics that are needed, right? Okay, and then what happens? Next up is myelination of the neurons, or myelinogenesis. So the myelin sheath, we've covered this already in earlier chapters. It helps speed up nerve transmission by insulating the axon to keep the signal traveling down the axon, jumping from node to node. This is saltatory conduction we've covered before. Two kinds of glial cells build the myelin sheath. The oligodendrocyte is working in the central nervous system to do this. Schwann cells do it in the peripheral nervous system, right? So they work a little bit differently. They have a different structure, these two different kinds of glial cells, but their jobs are effectively the same, right? They're going to create the myelin sheath. This happens at this stage, this later stage, right? After pruning. Why are we doing this? Why do we wait until such a late stage of development to start myelinating axons? Well, let me be clear about a couple of things. Myelination occurs in stages, and some of the neurons of the peripheral nervous system, so these Schwann cells, um, build myelin sheaths in the peripheral nervous system pretty early on, even prenatally. It's the cortex that waits, right? So we're going to wait until we're a year, 2 years, even 3 years before we fully myelinate the whole of the cortex in the brain. Why do we do this? Why do we wait to do that? Because we think, oh, there must be some advantage to myelinating, right? Because if it speeds up the nerve signal traveling down the axon, shouldn't we want to do that early on so we can speed up processing? Well, there's a trade-off, as it turns out. So what we're dealing with here is the idea that yes, we want to speed up nerve transmission, but we also want to maintain plasticity, right? We want to continue to have some degree of neuroplasticity in the brain in these early years for as long as possible, because we want to make sure that we have all of these experiences. And sometimes experiences happen in our first year of life that help keep certain synapses active. But there might be other experiences that happen a little bit later, right? Especially as a child starts to develop some of their motor skills. So then what's going to happen is that we're going to um, we're going to start maybe fine tuning some parts of the brain in terms of pruning processes not until we're closer to 2 or 3 years of age, because the child's motor skills and cognitive abilities haven't yet kind of created the ability to form certain kinds of experiences, right? So there's going to be experiences that are happening to 2 and 3 year old children that would never happen to a 6-month-old baby, right? And what the myelin sheath does is that even though it speeds up nerve transmission, it also places some constraints on neuroplasticity. It places some constraints on how much an axon can further grow beyond that point. So we want to make sure that we don't do anything to interrupt neuroplasticity um, in those parts of the brain that are still developing and still growing, and where we still have some arborization and maturational processes that have to happen. So we're going to let all of that happen. And then once the experiential stuff has mostly done, then we can start myelinating the cortex, right? Because then we're going to say, okay, we know which neurons, which synapses are useful, and we're going to keep them. Which ones are not, we're going to get rid of them. And only once we've done all of that can we then start to invest this energy into myelinating the axons that are left over. Okay, next up is we're going to talk about research in early childhood and how this affects how we can understand the relationship between experiences and specific developmental processes. But I think this is yet another good place to stop the video at this point. So we're going to stop it here. We're going to come back and talk about doing research in early childhood brain development to finish up. I think in one more video, I think will be enough to finish up this particular chapter here.